So my goal today is to tell you about some of the work that we've been doing focused on tuberous sclerosis and really using tuberous sclerosis as a potential inroad to understand autism. Let me start with the problem that we're all facing in the autism field right now, the amazing heterogeneity of genetic causes that give you susceptibility to developing autism. So the blossoming of research in genetics and genomics has now told us that there are probably 400 to 1,000 different susceptibility genes for developing autism spectrum disorder. So that's been wonderful in terms of gene finding studies and publications in this area. It's apparently a major obstacle when it comes to actually providing new treatments for autism spectrum disorder. And that's really the main focus of my studies for the past decade or so. So let me tell you about some scenarios that I see coming up. One can imagine a scenario where we have a broad spectrum treatment that works for all forms of autism. That's an optimistic scenario. I think some of the work that's coming out from labs such as Mark Baer suggests that the synaptic abnormalities that occur in subtypes of autism, such as tuberous sclerosis and Fragile X, might be quite different from each other. And in fact, 180 degrees opposite from each other. So you can imagine a scenario where what works in Fragile X may actually be harmful in tuberous sclerosis and vice versa. And on the other side, we have this more pessimistic scenario of having to develop a different drug or different intervention for each different genetic cause of autism. And I don't think we have the resources for that scenario at this point. So many of us in this field are hoping for somewhere in between where there might be subcategories of autism in which there's convergence either at the cellular level or at the circuit level. So a treatment that works for one disease may work for a number of diseases that share the same convergent pathways. So what I'd like to argue today is that tuberous sclerosis might be one of those pioneer diseases that defines a subcategory of autism. So at Boston Children's, we try to take advantage of the patients that we see. And we usually start our studies with genetically defined autism spectrum patients. We try to do as deep phenotyping of these patients as possible using advanced EEG, MRI, and biochemical techniques. We have used cell-based models. In the past, most of these were mouse or rat models in culture. But more and more, we are using human iPSC-derived neurons in cell culture. We have moved on to animal models, and I'll show you some of our work on a conditional knockout of TSC. And we've done some treatment trials in the setting of those animal models that have led to clinical treatment trials. As in Fragile X and Rett syndrome, I think we are going to go through this cycle a number of times to come up with effective and safe treatments for these disorders. Let me tell you about why we started studying tuberous sclerosis almost 15 years ago now. 50% of the patients with TSC are affected with autism spectrum disorder, so that's a high ratio. But very importantly, many of the TSC patients will be diagnosed before birth or at the time of birth because of the cardiac lesions that these patients develop in the newborn period. I'd like to argue that the cellular mechanisms aberrant in TSC, the mTOR pathway that I'll tell you about today, are beginning to be understood, not just from the neuroscience perspective, but from studies in cell biology, oncology, transplant medicine, et cetera. And then, very luckily, we have FDA-approved specific inhibitors of the mTOR pathway that can be repurposed for preclinical or clinical trials. So the combination of these four factors, I think, makes tuberous sclerosis a unique opportunity in the autism spectrum field. So many of you are familiar with tuberous sclerosis, but just to remind everyone, it causes benign tumors in the brain, eye, skin, kidneys, and the heart. It usually presents to child neurologists like myself because 90% of the patients will develop seizures sometime in their life. About half of them are affected with intellectual disability and autism spectrum disorder. The incidence is about 1 in 6,000 to 1 in 10,000 newborns. So in the United States, we anticipate there are about 50,000 people affected with tuberous sclerosis, and in the world, about 1 million people affected with this disorder. The genes have been known for about 2 decades. TSC1 that encodes for hamartin and TSC2 that encodes for tuberin are the causal genes. And loss of function mutations in one copy of these genes is sufficient to have tuberous sclerosis. So more and more we diagnose TSC with MRI scans. So this is a FLAIR image of a patient with tuberous sclerosis. They have these cortical dysplasias, also called cortical tubers. On average, a patient with tuberous sclerosis will have 20 to 30 cortical tubers. If you look at the autopsy specimen of a patient with tuberous sclerosis, you'll see that these cortical tubers are associated with thickening of the cortex and lack of myelination underneath that tuber. If you look at that area under the microscope, you'll see the presence of these giant cells, which are about 250 to 300 microns in diameter, about 10 times the size of a normal neuron. And any organism and any cell that's missing the TSC1 or TSC2 genes actually grows out of control. Here's a Drosophila. This is the wild type eye of a Drosophila. This is a TSC mutant. The whole eye is bigger. Ommatidia that make up the Drosophila eye are bigger. And the hair cells are bigger when they're missing the TSC gene. So why does this happen? It happens because of a very well-conserved pathway in cells that's regulated by this GTPase called Reb, and this kinase called mTOR, which stands for mechanistic target of rapamycin. So when these two proteins are active, you turn on protein synthesis, and the cell grows. And what TSC1 and TSC2 proteins do is to form a complex that puts a brake on Reb. In fact, TSC2 is a GTPase activating protein or inhibitor of this GTPase Reb. So if you're missing either TSC1 or TSC2, this complex becomes dysfunctional. You have too much activity of mTOR, resulting in too much cell growth. Cells have taken advantage of this pathway in several ways. So growth factors such as insulin or BDNF activate a kinase called AKT that phosphorylates TSC2 on a certain set of residues and inactivates this complex that allows mTOR activity, resulting in protein synthesis and cell growth. So growth factors inactivate TSC2 complex and result in cell growth. Conversely, energy level in the cell does the opposite. So if you break down your ATP to AMP, AMP activates a kinase called AMP kinase, which phosphorylates TSC2 on a different set of residues and actually activates that complex. That puts a brake on mTOR and puts a brake on protein synthesis, so a starving cell doesn't continue to grow. So one of the first questions that we got interested in when we started studying tuberous sclerosis was, what is the true incidence of autism in the TSC population? If you looked at retrospective cohort studies in the literature, it ranged from 17% to 80%, depending on what cohort you were studying. So we did a prospective study together with Patrick Bolton in the United Kingdom. And Shefali Jesti, who was a fellow with us at the time, actually looked at data that was collected in the United Kingdom of infants born with TSC, who were followed with yearly examinations, including ADAS, till 5 years of age. So what Shefali did was to analyze this data with Patrick's group, and what she showed was that about 20% of the patients had full-blown autism at the time, and another 30% were on the autism spectrum disorders. The other 50% were not on the spectrum. So this ratio of about 50% autism spectrum disorder toll has held up in other prospective studies since then. So we think the true incidence is about 40 to 50% of the patients born with TSC. So one question I get asked a lot is the autism due to TSC the same as autism in the non-syndromic population? So in my practice, when I see a patient with tuberous sclerosis and autism walk into my office, if I don't notice the skin lesions that they have, I can't tell if they have tuberous sclerosis or not. Their behavioral manifestations seem very similar to those in idiopathic autism. But that's just anecdotal. There have been actually 2 studies that have looked at this question. So the first one was from Patrick Bolton's group again, where they use ADI interview analysis of autism symptoms in several genetic disorders and compared them to those of the AGREE sample. And they did a principal component analysis of this ADI subscores. And the genetic disorder that matched the closest to the AGREE sample in terms of ADI subscores was tuberous sclerosis. More recently, Shefali Jesti collaborated with us and looked at patients with TSC, with no autism, non-syndromic autism, and TSC patients that are on the spectrum. And she looked at the ADAS subscores of these patients. And as you can see, the TSC patients shown in this purple line with autism and the non-syndromic autism patients look very similar to each other in terms of symptom severity and symptoms. So I think both from the ADI and the ADAS analysis, autism in TSC looks very much similar to what we see in the non-syndromic population. So why do individuals with TSC go on to develop autism? Much of the earlier work focused on the cortical dysplasias or cortical tubers. There was a study that suggested that having bilateral cortical tubers in the temporal lobes were necessary, but not sufficient for developing autism. Unfortunately, this was done with 1.5 Tesla MRI. More and more, we are doing 3 Tesla MRI, and almost every patient with TSC will have some lesion in the temporal lobe. So that correlation doesn't seem to hold very much. And there have been several other cohort studies that failed to show a similar correlation with the temporal lobes, but implicated the cerebellum or the frontal lobes. So I think this relationship between cortical dysplasia and autism is still up for investigation. In my practice, if I look at the conventional MRI of a patient with tuberous sclerosis, I can't tell which one is going to have autism, which one is not going to have autism. So here's an example. This is a 17-year-old boy with these 3 cortical dysplasias in the frontal lobes. He has mild learning disabilities and is an integrated class in high school. Here's an 18-year-old girl with similar lesions in the frontal lobes. She's nonverbal and autistic. So these conventional MRIs don't teach us that much, at least in my experience, about the severity of involvement in terms of social symptoms. So instead of focusing on these white spots, we've decided to focus on the non-tuber pathology of tuberous sclerosis. And we've been doing a lot of diffusion tensor imaging, which I'll tell you toward the end of the talk. So human studies are difficult to draw conclusions from in terms of mechanisms underlying the behavioral manifestations that we're studying. So we've turned to mouse models for the most part to interrogate the hypothesis that TSC1 and TSC2 regulate neuronal connectivity and function through the mTOR pathway. What I'd like to do is to summarize some of the work we've done over the last decade or so in various aspects of this disease. So we basically allowed the disease and the TSC complex to take us in different directions in terms of neuropathology and neural connectivity. We did some work on axon guidance, mitochondrial dynamics, myelination, cerebellar circuitry, epilepsy, and circadian rhythms. For the purpose of this talk, I'm not going to get a chance to talk about epilepsy or the circadian rhythms, which are very important problems in tuberous sclerosis, but I'll focus on the rest of the other topics that we touched upon over the years. So to start on axon specification and guidance, when we started this work, it was clear that TSC1 and TSC2 proteins are found in the growth cones of neurites in culture. But it wasn't clear whether these neurites were dendrites or axons. So we took antibodies to the TSC-mTOR pathway and stained embryonic hippocampal neurons in culture. So here's a embryonic rat neuron in culture. Here are the short dendrites and the long axon, stained with TUJ1. When we stained the same neuron with a phosphorylated form of TSC2, as you can see, most of the staining is in the distal axon, and very little staining is seen in the dendrites. And this seems to hold true for Reb and S6 kinase as well, so other components of the mTOR pathway. So why do these TSC-mTOR pathway components, why are they enriched in the developing axon? So one of the earliest things a developing neuron has to do is to determine which one of its nascent neurites will become the axon. In the very beginning, each of these neurites have equal potential to become the axon. Over the next 48 hours, one of them grows faster than the others, and that becomes the axon. And the other ones become dendrites. If you physically ablate this axon, then one of the other neurites becomes the axon. So the developing neurons have an intrinsic program to make sure they have an axon and only a single axon. So we took advantage of this pathway that were studied by Gary Banker, Carlos Duddy, and many others, and we asked, what is the role of TSC-mTOR pathway in this process? So we stained hippocampal neurons in culture with a dendritic marker, MAP2, and an axonal marker, tau1. And we did this for control neurons and TSC-deficient neurons. We knocked down TSC2 using shRNA, or we knocked out TSC1 using a Cre-Lux system. And we saw exactly the same results. So in the control neurons, we saw a single tau-positive process coming out of the cell body in the vast majority of the neurons. In TSC-deficient neurons, we saw multiple tau-positive processes coming out of the cell body, or multiple axons. And this was a very robust phenotype. When we quantified, in the control case, we saw 80% of the cells having a single axon. In TSC-deficient neurons, 40% of the cells had a single axon, and 40% had multiple axons. And importantly, if you treated these TSC-deficient cells with an mTOR inhibitor, such as rapamycin, we could reduce the multiple axon phenotype. So we asked, what is mediating this effect? We took advantage of a set of proteins that were described as being important in axon development, and we tested if their levels were modulated by TSC loss. One of those proteins, SAD kinase, turned out to be such a protein. So we are looking at Western blots of SAD kinase. As you can see in TSC-deficient cells, the SAD kinase protein levels go up significantly compared to controls. And if you treat those cells with rapamycin, the SAD kinase protein levels come back to controls. At the same time, we did not see any change in the mRNA levels of SAD kinase. So what's happening is that in the absence of TSC, in an mTOR-dependent fashion, a subset of proteins are being upregulated. One of those turns out to be SAD kinase, which promotes axonogenesis. So to test if SAD kinase was playing an important role in multiple axon phenotype, we knocked down SAD kinase at the same time we knocked down TSC, and we could significantly reduce the multiple axon phenotype. So we think that there's a program of protein synthesis that's regulated by TSC and mTOR in developing cells. We think SAD kinase is one of those mRNAs that's regulated by the mTOR pathway. We are now taking a more unbiased approach to identify those mRNAs that are regulated by TSC and mTOR in developing neurons and trying to identify other factors that might contribute to this phenotype. I should say now that this multiple axon phenotype that we see in TSC deficient neurons that I described only happens in cells that are null for TSC1 or TSC2 expression. But TSC is an autosomal dominant disease, and the vast majority of the neurons in the brain of a patient with TSC are missing only one copy of TSC1 or TSC2. So we asked, are there other abnormalities in axonal connectivity? And we turned to axon guidance. So axons navigate to their final destination in the central nervous system by interacting with either attractive or repulsive cues. Some of these are secreted molecules, others are membrane-associated molecules. And this has been studied by many labs over the years. One of the easiest and most well-studied pathways in the CNS in terms of axon guidance are the projections from the retina to the LGN, that lateral geniculate nucleus in the thalamus. The advantage of this pathway is that you can put different colored dyes into each eye. It's very easily accessible. It's been studied by many groups, and the axons make a topographic map in the LGN, and you can use this quantitatively to test if the connections are being formed properly. And very importantly, the molecules that are important for this topographic mapping has been identified by other groups. So EPH receptors on the retinal ganglion cells interact with repulsive ligands in the thalamus called ephrins, and this repulsive interaction is important for the proper mapping of these axons. So we use this well-studied system to ask, are there deficits in the TSC deficient mice? So we compared control litter mates to TSC2 heterozygous mice. So we put a small dye crystal in the corner of the retina, and we mapped those axons in the lateral geniculate nucleus. As you can see, in the wild type case, all these axons go to this corner of the LGN. When we did the same experiment in mice that are TSC2 heterozygous, the axons definitely travel to this expected location, but they also projected across the axis of the LGN. This is summarized in this cartoon here. So the wild type axons go to this area, and the TSC2 heterozygous axons end up across the axis of the LGN. So missing just one copy of TSC2 gives you exuberant and inappropriate connections in the lateral geniculate nucleus coming from the retina. Why does this happen? Well, to make a long story short, we did a single cell assay using growth cone collapse. And we showed that these axons that are missing one copy of TSC2 were less likely to be collapsed in the presence of the repulsive ligand, the ephrins. The wild type axons collapse about 70% of the time. TSC2 deficient axons only collapse about 30 to 40% of the time. So they seem to be less vulnerable to ephren stimulation. So why does this happen? So we did a comprehensive cell signaling analysis of ephren stimulation in neurons. And what we found was that if you put ephren, this repulsive ligand, onto cultured neurons, within 2 minutes of stimulation, you turn down the mTOR pathway. This is phosphorylated form of S6 kinase, which is one of the substrates of mTOR. And S6 kinase phosphorylates ribosomal protein S6, and that phosphorylation event also goes down in a time-dependent fashion. This happens at the same time ERK phosphorylation is downregulated in this really rapid fashion in cultured neurons. So we postulated that there might be regulation of the ERK pathway through this RAS gap downstream of EPH receptors. But the really functionally, the very more interesting question to us was, if the mTOR pathway is regulated by the EPH receptor activation, is this happening locally in the axon, or is it happening in the cell body? So to answer that question, we used a compartmentalized chamber. We plated the neurons on inserts that have small holes. So the neurites go through these holes, but the cell bodies cannot go through the holes. So you can scrape off the cell bodies, and you're left with a neurite-only preparation. So onto that preparation, we added S35-methionine to be able to quantify de novo protein synthesis. And we added the ephrin ligands as well as rapamycin onto this preparation. What we saw was that de novo protein synthesis in the neurites is downregulated by about 20% by rapamycin treatment. I'm sorry, 20% by ephrin treatment and 30% by rapamycin treatment. So this suggests that EPH receptors and the mTOR pathway can regulate de novo protein synthesis locally in the neurites. So here's our working hypothesis. In the presence of growth factors such as BDNF, you turn on the PI3 kinase AKT pathway and the ERK pathway, both of which together inhibit TSC2 complex. That activates mTOR and protein synthesis, resulting in growth and elongation of the neurite. But if you have the repulsive ligands, like ephrins, through double inhibition via ERK, you activate the TSC complex. You activate the brake. That reduces mTOR activity, that reduces protein synthesis, and contributes to retraction and collapse response. And of course, in the disease states, when you don't have as much TSC complex, even though the ephrin stimulation activates the EPH receptors, that does not communicate to the mTOR pathway. So you get exuberant growth and collapse resistance. So this has been our working hypothesis, and we've been testing this hypothesis in various ephrin-dependent pathways in the brain. So far, what I've told you so far really focuses on protein synthesis and translation. But it turns out that mTOR does a number of things in the cells. It regulates processes, anabolic and catabolic processes that are outside of protein synthesis. So the next aspect of mTOR pathway that we've been studying more recently is mitochondrial dynamics. Obviously, mitochondria have been implicated in autism in multiple studies. In this case, we focused on the role of TSC-mTOR pathway on mitochondrial dynamics in the axons. So the first evidence or suggestion that mitochondria might be abnormally regulated in the TSC neurons came to us from some of our cultured neuron studies. As you know, mitochondrial biogenesis and mitochondrial degradation are in a very well-regulated equilibrium. What we saw in cultured neurons was that there was an increase in autophagic vesicles in TSC-deficient neurons compared to wild-type neurons. We saw an increase in mitochondria itself in another knockout of TSC, in this case, a Nestin Cree knockout of Tuberous Sclerosis 1 gene. And this was a significant increase per cell. There was, in some sections, we saw almost a 10-fold increase in the number of mitochondria that were accumulating in these cell bodies. And then another group from Europe actually studied cortical tubers from TSC patients that were taken at the time of autopsy, and they saw these dense core vesicles and lysosomal accumulation in TSC-deficient epilepsy tissue. So this suggested to us that there might be some abnormality in the dynamics and homeostasis of mitochondria. So we wanted to ask the question, how is TSC1, TSC2 deficiency impact mitochondrial homeostasis in neurons? Was this happening through autophagy? Could this be happening through biogenesis abnormalities? So the first question that my postdoc, Darius Ibrahimi-Fakhari, did was to stain cultured neurons with mitotracker that stains mitochondria. And then he did FAC sorting to look at the intensity of mitochondrial dye staining in these neurons. So in this case, you are looking at predominantly in the cell body in terms of the mitochondrial intensity. So compared to control neurons, Darius saw almost a 2.5-fold increase in the mitochondria in TSC-deficient neurons. So this was happening in the cell body. How about the neurites? So Darius used different dyes to look at mitochondria in the neurites. And in this case, it's a MitoDS red staining in the neurites. And what he saw in the neurites, unlike the cell bodies, was that there was actually a decrease in the number of mitochondria per length, a decrease in mitochondrial density in the TSC-deficient cells. So unlike the cell body, there seems to be a reduction. This happened in vitro, but how about in vivo? So we did some EM on the corpus callosum of TSC-deficient mice, in this case, synapsin Cree TSC conditional knockouts. And we saw a small but significant decrease in the number of mitochondria in the corpus callosum axons as well. So are these mitochondria functional? Darius turned to a seahorse assay to look at mitochondrial respiratory chain function. And he saw that there was a reduction in TSC-deficient mitochondrial respiratory chain function in TSC-deficient cells compared to control cells. So even though the mitochondrial numbers seem to be increased in neurons, they are not as functional. Again, this is predominantly happening in the cell body. How about the neurites? So there are different dyes you can use. He used a dye called TMRE and another dye called JC1, and they both can essentially quantify mitochondrial membrane potential. And what he saw was that using either system, there was a reduction in TSC-deficient neurons compared to control neurons in terms of mitochondrial membrane potential. So these are rat neurons. We turned to human iPSC-derived neurons. So we took patients with 2 different mutations in TSC2. We also made a CRISPR knockout of TSC2 to make a homozygous knockout of the same gene. And we looked at the mitochondrial membrane potential in heterozygous or homozygous cells. And basically what we saw was a gene dosage dependent reduction in mitochondrial membrane potential in the absence of TSC. So heterozygous cells had lower mitochondrial membrane potential, and homozygous cells had even lower membrane potential. And this was true both for the cell body as well as the neurites. So just to summarize so far, we see an increase in the number of mitochondria in the cell body, whereas we see a reduction in the number of mitochondria in the axon. But in both cases, there seems to be dysfunction of the mitochondria. What could this be due to? Well, there are a couple of possibilities. One could be degradation of mitochondria is abnormal. Another possibility, the transport of mitochondria might be abnormal. That could account for this discrepancy between the cell body and the axon. So we first looked at autophagy, or mitophagy, the eating of mitochondria in the neurons. This is a busy slide, but it's a very simple concept. What Darius did was to look at the co-localization of mitochondrial markers, in this case, mitotracker, with autophagy markers, LC3. And what he saw was that compared to wild-type cells, there was a reduction in the number of LC3 positive mitochondria. So it seems like there's impaired recruitment of autophagosomes to the mitochondria in TSC-deficient axons. He also looked at the transport of mitochondria. So he did live imaging of mitochondria in axons. And as you can see, you can detect the movement of mitochondria in anterograde or retrograde directions using this live imaging. Many of the mitochondria are stationary, as shown with these straight lines. When he quantified this, what Darius saw was that there was an increase in the number of mitochondria moving in the retrograde direction toward the cell body that could potentially contribute to the increased number of mitochondria in the cell body and a reduction in the axons. We also saw this really interesting phenomena of what's called oscillating mitochondria. I hope you can appreciate this sort of zigzagging that happens. Mitochondria are moving back and forth. That's seen that's been described at least in neurons that are missing pink and Parkin, these Parkinson's disease genetic mutations that have been associated with mitochondrial deficiency. So one of the things that mitochondria do in axons is to be present at the presynaptic terminal and provide ATP and calcium homeostasis for the presynaptic function of neurons. This has been studied extensively by several groups, including Frank Pelosi at Columbia. So we collaborated with Frank's lab, and we got some tools to look at the location of mitochondria in the axons in our TSC-deficient cultures. So we looked at the co-localization of mito-DS red for mitochondria with V-glut venus for presence of presynaptic terminals. And what we saw was that there was a reduction in the number of presynaptic mitochondria, or presynaptic puncta with mitochondria in TSC-deficient neurons compared to control neurons. So this suggests, again, to summarize, not only is there a reduction in the number of mitochondria in the axons, but there's also a reduction in the number of presynaptic mitochondria. We hypothesized, and I don't have time to get into the details, that this deficiency was occurring due to abnormal or deficient mitophagy that was happening in the axons. So we hypothesized that if we could enhance autophagy in TSC-deficient neurons, we could partially rescue this phenotype. So Darius took 2 different compounds, rapamycin, to inhibit mTOR, which is known to increase autophagy, and carbamazepine, which is an anti-seizure drug, but is known to enhance autophagy in an mTOR-independent way. And he treated his neurons, TSC deficient and control neurons, with these two compounds, as well as a combination of these two compounds. And in both cases, he showed that you could increase the number of autophagy markers that co-localize with mitochondria. So he could enhance autophagy and mitophagy using rapamycin and carbamazepine. Then he looked at the number of presynaptic mitochondria. And either rapamycin or carbamazepine or the combination of these two drugs could enhance the number of mitochondria that was associated with presynaptic puncture. So here I think is one case that we've shown that TSC deficiency and mTOR hyperactivity can result in abnormalities in neurons aside from aberrant protein synthesis. Mitochondrial homeostasis and mitochondrial transport also seem to be affected with this deficiency. And we think they might be partially contributing to the at least the presynaptic and potentially postsynaptic phenotype that's seen in TSC deficiency. So these two stories I told you about on the axon specification and mitochondrial dynamics both really highlight the cell autonomous abnormalities that we see in TSC deficiency. But we're starting to start to understand the non-cell autonomous effects of TSC loss in neurons as well. And I'll tell you one short story about that aspect, and that focuses on myelination that Jackie mentioned in her introduction. So we stumbled onto this almost 10 years ago, almost by accident, I guess I should say. We were collaborating with David Kirotowski's lab at Brigham and Women's Hospital, and he made a knockout of TSC under the synapsin Cree promoter. So if you delete TSC1 or TSC2 gene in all the cells of the body, mice die embryonically, so it's very hard to study them from a neurological perspective. And so we have to use conditional knockout models of TSC1 and TSC2. So this conditional knockout of TSC2 in just postmitotic neurons develops seizures. They are born at the expected frequency, but develop seizures around 3 weeks of life. And they die from seizures by P60. If you look at the brains of these mice, they don't show cortical dysplasias, but they show a marked hypomyelination phenotype. So this is myelin basic protein staining in the cortex from P7, P14, and P21. As you can see, there's increased amount of myelin deposition over the first 3 weeks of life. This is the control litter mates, and these are the TSC1 deficient litter mates. As you can see, there's reduction in both the gray matter and the underlying white matter in terms of myelin basic protein staining. So the first thing we did was to develop a paradigm to treat these mice with mTOR inhibitors. We used both rapamycin and everolimus. It's also called RAD001. We identified some pharmacokinetic factors that told us what the right dosage was, and we started treating these mice at P7 around this time, much earlier than when the seizures started. So vehicle-treated mice all died from seizures, as I told you, by P60. If you started treatment by P7, 90% of the mice survived, and they did not display any seizures. If you stopped the treatment with rapamycin at P30, about a month later, they started having seizures, and they started showing early mortality. So maintenance of mTOR inhibition seemed to be very important, and we saw almost identical results with everolimus. You may ask what happens to myelination. So this is the control brain, this is the mutant brain, and this is the mutant brain on rapamycin. Much of the myelination comes back to control levels. So in this case, remember, the cells that are putting down the myelin sheet are oligodendrocytes that are wild type for TSC1 expression. The cells that are deficient in TSC are the neurons. So what's happening to these myelinating oligodendrocytes? Why are they getting affected with loss of TSC in the neurons? So we started working on this approximately 7 years ago, and finally we were able to publish this paper. So I'll take you through this paper relatively quickly. So we first stained the mutant brains with CC1, a marker of mature oligodendrocytes. And as you can see in the mutant case, there's a significant reduction of these green cells. It's quantified here. There's a reduction both in the gray matter and in the white matter. And if you treat the mutants with rapamycin, you can essentially rescue the reduction in the oligodendrocytes. So this seemed to suggest to us that there was some communication between the TSC deficient neurons and the maturing oligodendrocytes. We hypothesized that this could be happening through a secreted factor. So we took conditioned media from TSC deficient neurons in culture. We concentrated that conditioned media, and we placed it onto oligodendrocyte precursor cultures. And we stained the OPC cultures with stage-specific markers to ask how are these oligodendrocytes maturing in culture in the presence of TSC deficient conditioned media. I won't show you all the data, but I'll show you one of the important findings. So we looked at myelin basic protein staining as a marker of mature oligos. Compared to control condition media, TSC deficient condition media from neurons significantly inhibited the MBP staining in these oligodendrocyte cultures. We also did this with earlier markers of oligodendrocyte maturation. In those cases, we actually saw an increase in the staining. So what we think is going on is that there's a block from immature pre-myelinating oligodendrocytes to myelinating oligodendrocytes when the cells are treated with TSC deficient condition media. So we asked what in the TSC deficient condition media is responsible for this effect. We did an expression profile of the TSC deficient cells, and we focused on secreted molecules. On the top of the list, near the very top of the list, was a growth factor, or it's called a growth factor. CTGF stands for connective tissue growth factor. It's been implicated in myelination in other studies as well. So we wondered, is CTGF the molecule that's mediating this effect? So first of all, is CTGF expression truly increased in TSC deficient cells? By Western blot, it's increased, as you can see here. CTGF secretion is also increased TSC deficient condition media by quantifiable Western blotting. These were, again, in rat neurons. We also turned to human neurons. So we took iPSC-derived neurons from TSC patients. And there's almost a twofold increase in CTGF expression in human neurons as well. So we returned then to our condition media experiment. As I had told you before, if you treated these oligodendrocyte precursor cultures with TSC deficient condition media, you can reduce the number of cells that express myelin basic protein. What happens if you treat the cells with condition media in the presence of an antibody to CTGF to neutralize the CTGF effect? So we added that antibody, and we could partially but significantly reduce the effect on MBP staining. So this suggests that CTGF, at least in culture, is mediating this effect. But how about in vivo? So to get to that question, we made a double knockout. We knocked out TSC in neurons, but we also made a double knockout of CTGF with TSC1. So synapse-increased CTGF TSC1 double knockouts had essentially normal levels of OLIG2, in this case, a maturing oligodendrocyte marker. As you can see, both in the gray matter, we normalized the oligodendrocyte number, and we could rescue, but not completely rescue, the oligodendrocyte number in the double mutant in the white matter. So these studies suggest to us a few things. One, that there are non-cell autonomous effects of TSC loss in neurons, and we need to start to understand these better. It seems like CTGF is an important factor of myelination, in TSC at least. We're starting to look at other diseases of myelination as well. And thirdly, that myelination can be modulated, even in mouse models that have severe myelin defects, by mTOR inhibitors. And myelination, as you know, is a relatively easy thing to measure using MRI in patients. So I'll come back to potentially using MRI as a tool to be able to look at TSC deficiency in the brains of individuals affected with tuberous sclerosis disease. Now, the final aspect I want to talk about is cerebellar circuitry. So one of the, I think, major pitfalls in the autism field is that we don't really know what cell type or what region of the brain where the underlying pathology is residing. Unlike ALS or Parkinson's disease, where we sort of have a pretty strong sense of which circuits are affected. In autism, that is still a mystery. So we took an approach where we started deleting the TSC1 or TSC2 genes in different cell types or different regions of the brain. We've done a number of those conditional knockouts now. The one that's been the most informative to us has been the loss of TSC in the cerebellum. And why did we focus on the cerebellum? Well, there's a number of studies that have implicated the cerebellum. Some neuroimaging studies have shown that in individuals affected with autism spectrum disorder, there's decreased vermis volume and abnormal white matter in the cerebellum. There have been a handful of pathologist studies done by several groups, but it seems like one of the most consistent finding in those pathology studies has been a reduction in Purkinje cell number in individuals affected with autism spectrum disorder compared to typically developing individuals. And then a study from Kathleen Poropoulos when she was at Boston Children's showed that if you look at newborns with isolated cerebellar hemorrhage in the newborn period, about 37% of those individuals go into develop subsequent autism. So there's almost a 30-fold increase in the relative risk of autism in the individuals that have only hemorrhage in the cerebellum in the newborn period. So these are all sort of descriptive studies, obviously. So to be able to address the role of cerebellum in a more mechanistic way, we turned to mouse models. So we wanted to probe the hypothesis that cerebellar dysfunction could contribute to the pathogenesis of autism in TSC mice. We made a knockout of TSC1 gene on the Purkinje cells, the only output cell of the cerebellar cortex. So this work was done by Peter Sahin in the lab. He used L7 Cre as a way to delete the TSC1 only in the Purkinje cells. These mice have a normal lifespan. They have normal weights. They are relatively healthy compared to their litter mates. So we controlled 3 cohorts: control mice, wild-type control mice, heterozygous knockouts, and null mutants. And with Jackie's help, we used her 3-chamber apparatus and looked at the social approach. I'm showing you just a subset of the data. But as expected, in the control cases, we saw that the wild-type animals were interacting more with the animal than the object. But our heterozygous and null mutants did not show that preference. They spent equal amount of time with the animal as the object. And the time investigating is plotted here. They really show no significant preference for the animal than the object. We also looked at grooming and reversal learning, and they showed deficits in those as well. To be able to look at one objective measure of the abnormalities these animals were showing physiologically, we collaborated with Wade Regier and recorded from Purkinje cells that are missing the TSC1 gene. What we saw was that there was a gene dosage-dependent decrease in the excitability of these Purkinje cells. So the heterozygous Purkinje cells fired less than control Purkinje cells, and null Purkinje cells fired even less. So our first question was, can we rescue this defect using an mTOR inhibitor? So we turned to rapamycin again, and we started treating these mice from P7, essentially as soon as the Cre gene is turned on. And what Peter saw was that in the vehicle-treated case, we saw exactly what I showed you before. The animals spend equal amount of time with the animal as the object. But the rapamycin-treated mice spend much more time with the animal than the object, essentially normalizing to what their litter mates show when they are treated with rapamycin. So early treatment with rapamycin could rescue this defect. Now, this is all in mice, and obviously the question that we are particularly interested in, do we see similar abnormalities in human cells? So for the past several years, we've been trying to make human Purkinje cells. So we've been collaborating with Mary Beth Hatton's lab at Rockefeller, and we developed a cell culture algorithm to make Purkinje cells from iPSCs. This protocol takes approximately 140 days, so it's extremely long. But the cells at the end develop markers of Purkinje cells that are recognized as PCP2 expression, GAD, and GABA receptor expression. And if you do cell sorting, you can end up with approximately 60 to 80% pure Purkinje cells in culture. So we've been using these cells to record from them and ask, what is their excitability like? So this was a collaboration with Wu's lab. We've been doing patch clamping. And what we see is very similar to what we see in the mouse Purkinje cells. So TSC2 heterozygous Purkinje cells fire less frequently than wild-type Purkinje cells. The null cells fire even less. And rapamycin treatment partially rescued this phenotype. So we're really excited about the fact that we now have a human cell culture model for at least Purkinje cell development in the absence of TSC. And some of the electrophysiological abnormalities that we're seeing in cell culture look very much similar to what we see in the mouse model. So we would like to use this cell-based platform to be able to do some screens for identifying molecules that could rescue this phenotype. So just to summarize, I've described some of the work we've done in the mouse models. Obviously, the main question we're interested in, how can we help our patients with tuberous sclerosis and autism? So in the autism field, one of the prominent theories is that there might be developmental disconnection between the regions of the brain that control social interaction, repetitive behaviors, and language in those individuals with autism compared to typically developing individuals. The one advantage that we have in the TSC field is that we can identify individuals with TSC very early on in life. So here's an echocardiogram of a newborn, and the presence of these cardiac tumors allows us to identify these patients very early in life, in many cases prenatally these days, or at the time of birth. There was a study done by Wayne Turetsky a few years ago where he looked at fetuses and newborns with multiple cardiac tumors, and the chance of having TSC in that cohort was 95%, which is essentially better than genetic testing. So we know that these patients have a 95% chance of developing tuberous sclerosis, and among TSC population, there's a 50% chance of developing autism. So we've been asking a very simple question. Can we detect which infants born with TSC will go on to develop autism? And this has been a collaborative, ongoing study with Chuck Nelson and Simon Warfield, as well as Shafal Jesti. So we've been doing neurocognitive assessment of infants, diffusion tensor imaging, and neurophysiological assessment of face processing. I'll show you one piece of data from this work. This was diffusion tensor imaging of the corpus callosum as a three-dimensional region of interest. We compared 3 different groups, typically developing children, TSC patients that are not on the spectrum, and then finally TSC patients with autism. And what we saw was that these 2 groups were really not very different from each other, as you can see visually, but also by fractional anisotropy values shown here. They were nearly identical. But TSC patients on the spectrum had reduced fractional anisotropy and actually increased radial diffusivity of the corpus callosum. Now, radial diffusivity suggests that there's lack of organization of the myelin in the corpus callosum. That's at least one of the factors that's been associated with reduced fractional anisotropy and increased radial diffusivity. This suggests to us that the microstructure of the corpus callosum in individuals only with autism compared to those without autism is abnormal in TSC. So this gives us hope that even within a genetic disorder with a homogeneous genetic cause, such as TSC, we might be able to differentiate to those that are at high risk versus those that are at low risk with tuberous sclerosis. Now, this was done in a relatively old age group. Our goal would be at some point to, if when a newborn with TSC walks into our clinic, that we should be able to tell them using some marker which group their child falls into. And if they're in a high-risk group, we might be able to treat them differently than those who are in a low-risk group. So this is important for counseling purposes, but it's really only matters if we have something to offer in terms of treatment. So mTOR inhibitors, as I showed you earlier, look very promising in terms of mouse models. I showed you some data on myelination and seizures in our mouse model of TSC loss in neurons. Michael Wang's lab at WashU showed that he could prevent or stop seizures in an astrocyte-specific knockout of TSC. Alcino Silva's lab had a very influential paper showing that just 5 days of rapamycin treatment can improve learning and memory in a mild mouse model of tuberous sclerosis. And I show you some of our data preventing autistic features in the cerebellar knockout. So based on these studies, a number of years ago, we launched a phase 2 trial of an mTOR inhibitor, in this case, everolimus, in TSC patients. And we chose neurocognition as our primary endpoint. We enrolled 47 patients from 2 sites, Cincinnati and Boston Children's, between the ages of 6 and 21, and a IQ cutoff greater than 60. This was a randomized placebo-controlled trial. We looked at neurocognitive testing, including CANTAB, at 0, 3, and 6 months. And we looked at autism as measured by SRS and ADAS, seizures, frequency, and sleep disorder in these patients as secondary endpoints. So this study is recently finished, and unfortunately, we don't see an improvement in those treated with everolimus compared to the placebo-treated patients. So this is extremely disappointing, and I can spend some time, why we think that might have happened. But one of the things that we did not have in this study was any biomarker to detect any possible improvement. We really used neurocognitive assessment as our endpoint. So in order to overcome that deficit, we formed a consortium. It was an Autism Center of Excellence network that was funded by the NIH of 5 sites around the country that are geographically distributed. And we collected infants born with TSC. And we did standardized neuropsychological assessment, MRI, and EEG on these infants prospectively till they are 3 years of age, with the idea that we could identify potential red flags or biomarkers that could predict autism diagnosis at 36 months. We don't have all the data because not all the children have reached the age of 36 months yet, but we have some preliminary data that I'd like to share with you. So this was on the role of EEG as a potential marker of seizure onset. So we asked the question, does EEG predict seizure onset, and with what interval? So we looked at prospectively EEG in children and obtained it essentially every 6 weeks, every 3 months, and then every 6 months going to the 2nd and 3rd year of life. What we saw in some cases that the children had normal EEG and never developed seizures, as shown in this white bars here. But in other cases, what we saw is that they started out with normal EEG early on, and eventually they developed abnormal EEG, as shown with this gray bar, and eventually they had this diamond, which is the onset of clinical seizures. So when we quantified this, what we saw was that the average age of abnormal EEG was at 4 months, or 4.2 months, and the average age of first-time clinical seizures was around 6 to 7 months. So there's a nice 2.5 to 3 month period where the EEG is abnormal, but the clinical seizures have not started. And at least in this small cohort, the positive predictive value was 100%. Since then, we've done more infants, and the value is slightly less, but not much less. So this gave us the idea that we might be able to intervene in that 2 to 3 month period with pharmacological intervention to try to prevent the process of epileptogenesis. So we now have a R01 grant with Martina Beben as the PI, using the same network of now 7 sites across the United States. It's called PREVENT, for preventing epilepsy using vigabatrin in infants with tuberous sclerosis. And after discussion with the FDA, we chose not the onset of clinical seizures, but developmental impact of developmental impact at 2 years as our primary endpoint. And we're comparing early treatment versus late treatment with an anti-seizure medication called vigabatrin. Vigabatrin is a GABA transaminase inhibitor, and it's thought to increase GABA at the synapse. And it's been used for many years for treating infantile spasms in TSC patients very effectively. So this is a relatively safe and well-tolerated drug in this age group. So this study is just started, and we're enrolling patients right now. So if you have any infants with no seizures, they could be potentially recruited to the study. And I think the closest site is at Stanford with Brenda Porter. But I'm very excited about the idea of moving earlier in terms of prevention trials in these kind of neurodevelopmental disorders, where we can identify the risk factors early in life. So I've only talked about TSC1 and TSC2. And just to remind you, there's another 900 plus genes that are associated with increased susceptibility for developing autism spectrum disorder. So how are we going to get to this subcategorization? How are we going to identify subcategories that could potentially form groups of patients that we can treat with the same potential intervention? So to be able to get at that question, we've took some educated guesses of what disease groups can fall in the same category. And we focused on the mTOR pathway. We formed a rare disease clinical research network, or RDCRN, again funded by NCATS at the NIH and NINDS. It's called Developmental Synaptopathies Consortium, and it focuses on the mTOR pathway as a potential underlying cause of neurodevelopmental disorders. As a part of that, we're looking at 3 different genetic disorders that we hypothesize to be in the same pathway. So obviously TSC that I've been talking about, but PTAN, which is biochemically very proximal to TSC in terms of regulation of AKT and mTOR. And then SHANK3, or Phelan-McDermott group of patients, which are quite abundant and also show abnormalities that are affecting this pathway. So we have this consortium. We are collecting natural history study on 100 patients with TSC, 100 patients with Phelan-McDermott syndrome, and 100 patients with PTAN, hamartoma tumor syndrome. We're doing deep phenotyping of these patients, as well as looking for biomarkers using EEG and MRI. And we also have a pilot study of clinical treatment trial using everolimus in the PTAN patients. So this is at the clinical level, and this study is ongoing. To be able to translate effectively from mouse models that I showed you earlier to this developmental synaptopathies consortium, I've also gotten engaged, as Jackie mentioned, with the Autism Speaks as a part of their PACT consortium, which stands for Preclinical Autism Consortium for Therapeutics. This was a really, I think, forward-looking idea from Autism Speaks to be able to do standardized testing and electrophysiological assessment in mouse models of autism. And as a part of that, we've done now 6 different genetic disorders that have been associated with autism in the mouse models. And we've done deep phenotypical evaluation of those mouse models in Jackie's lab and electrophysiological assessment in our lab at Boston Children's. So I think this will start to give us a cross-diagnostic comparison of these disorders, giving us the ability to focus on similarities and differences between these disorders, and potentially make some predictions based on the mouse models to move on to clinical trials in the population of patients that we see in the clinic. So I'm very excited about overlapping the Autism Speaks PAC consortium with the Developmental Synaptopathies consortium, and really learning from each other, as we've done in the TSC population, going back and forth between what we see in the lab and what we are seeing in the clinic. So I'll stop here and acknowledge some of the people that did the work. So Alessia DiNardo did the work on axonal development. Darius worked on the mitochondrial homeostasis. Ebru Ercan was the lead author in the myelination. Peter Tsai did the work on the mouse model of TSC in the cerebellum, and Maria Sandwork developed with Mary Beth Patton's lab the Purkinje iPSC protocol. This is our packed group that works on the EEG in the mouse models with Jackie's lab. And then this is our iPSC core that's been led by Ivy Chen and Liz Buttermore. I should acknowledge many collaborators, including Jackie here, and our funding sources. Thank you very much.
What effects does rapamycin have on other tumor disorders like neurofibromatosis, Turcot syndrome, and things of that nature? Yeah, would it cure the tumors or any developmental concerns with those diseases?
That's a great question. So in NF1, for instance, there's some preclinical suggested that there are abnormalities in the mTOR pathway as well. I believe I'm not involved with them directly, but I think people have done some treatment trials using mTOR inhibitors in the tumors associated with NF1. I don't think they were successful to my knowledge. I think there's evidence that aside from the mTOR pathway, the ERK pathway is also very important in NF1 and other related disorders. So it might be that you may need combination therapy or some novel way of looking at downstream factors in those diseases. I think the advantage of TSC is that there's a very proximal relationship between TSC and mTOR, and that seems to be the major downstream effector pathway. Yes, David?
Mustafa, first of all, terrific talk, and it raised lots and lots of questions. I'll ask you some later on. But I was wondering, in terms of your clinical trials, so with the adults where you tried the mTOR inhibitor, it didn't produce a positive outcome, but you've now gone to younger subjects. Instead of using an mTOR inhibitor, you're going to a classical anti-epileptic. So just out of naivete, I'm curious, what are the hazards or the problems with using mTOR inhibitors in younger children?
That's a good question. Well, in general, we don't know as much about using mTOR inhibitors in young children as we know in adults. So rapamycin has been around for 2 decades, and it's been used in solid organ transplants, heart and kidney transplants for many years, and we know its side effect profile. So it's actually quite well tolerated, especially if it's used by itself versus with other immunosuppressants. You get oral ulcers, you get some abnormalities or high cholesterol and triglycerides. You have some amount of immunosuppression. You have, in very rare cases, you get this idiopathic lung disease that seems to be, for the most part, reversible. So overall, it's pretty well tolerated. There was a large phase 3 study of everolimus in seizures in TSC, and in that case, the side effect profile was quite favorable as well. So in the older population, we know those side effects well. We don't have as much data in the younger population, although we're getting more and more, because for the cardiac rhabdomyomas, the only treatment option in the past was surgical resection for those that didn't go away by themselves. And now several groups have used everolimus or rapamycin for those tumors, and it seems to help shrink them. There's a number of case studies in the literature, and we recently started a patient on that too. So I think we're going to get more and more data on how well tolerated they are in the younger group. In this case, vigabatrin was an obvious choice because it's been used for over a decade in this age group, and we have so much safety data in that it had to be the first choice to go with. But we've already submitted an application to the NIH to try everolimus or rapamycin in young infants that are at high risk. In this case, those that are detected by an EEG abnormality. So hopefully we'll do that one day.
Have any studies been done with TSC in middle-aged patients? And is there any way to prevent TSC for children or anyone else?
That's a great question. So there are actually a number of studies that have been done on the brain tumors and kidney tumors that TSC patients get, and those are the manifestations that we see more commonly in the older ages. There's also some studies that were done on cystic lung disease that women with TSC get. And all those three indications, FDA has approved mTOR inhibitors. So at least for those indications, mTOR inhibitors seem to be very effective. The question of whether we can prevent TSC altogether, I don't think we know of any way of doing that at this point. But hopefully we can at least prevent the manifestations of TSC, and I think our trial is one of the first to look at that.
Hi, thank you for the talk. I was particularly interested in one of the points you brought up early on about the size of the cells.
Yeah.
It's actually a phenotype we see in the temporal lobe in some of our young cases of autism in postmortem tissue. I just wanted to pick your brain on your thoughts of the functional implications of overlarge cells.
Functional implication of the large cells? So what we know so far is that those large cells have, for the most part, have second hits. So all the cells in an individual with TSC are missing one copy of TSC1 or TSC2. But it seems like a subset of cells, a relatively small subset of cells, stochastically, have a second hit. And those cells grow larger, and they probably contribute to these giant cells that I showed you earlier. Are they functionally contributing to the behavioral and neurological abnormalities that we're seeing? So that's up for debate these days. There's a pretty good correlation between where the tubers are, the cortical dysplasias are, and where the seizures come from. I think for the most part, it's thought that it's the perituber area where the seizures come from, not the actual tuber themselves. The tubers, some people have recorded from them, seem to be relatively electrically silent or not as active. So it's the area that's surrounding the tuber that seems to give rise to the electrical activity. And as I showed you some data, it's not clear how well the tubers correlate with the autism diagnosis. So I sort of think about the tubers or the giant cells being a modifier of the TSC gene. But I think a lot of the abnormality, as I've showed you hopefully, is due to the abnormal connectivity and abnormal function at the heterozygous regions probably. Yeah, hi.
I noticed that your research is mostly in tuberous sclerosis. As far as autism goes, is it that much of a cause for autism that you would, do your research for that as opposed to other causes for autism?
Right, that's a good question. So we estimate that about probably 1% of the autism patients overall have tuberous sclerosis. So there's probably 1 to 3 or 4% of autism patients that have Fragile X, and another maybe 1% with SHANK3 mutations or Phelan-McDermid mutations. So all of these genetic disorders that are relatively common probably each account for a couple of percentage points each, and altogether maybe account for 15, 20% at most of those highly penetrant genetic cause of autism. The reason we are focusing on TSC in this case, I think it provides us with a unique opportunity to identify these patients very early on. So infant SIP is the other group of patients that have been studied in depth, and the chance of having autism, a second child with autism, is probably on the order of 20% or so. In this case, we have we have a 50% chance of having autism, and we can identify these patients even before birth. So I think this provides us with a unique opportunity to go after a high-risk population, complementing the infant SIP studies that's been done with many groups.
So the axon exuberance data that you showed is very interesting in neuronal cultures, which is an interesting possible mechanism. If you have too many axons sprouting from a given neuron, obviously you're going to form incorrect connections.
Yeah.
So did you find a rescue of the multiple axons with any rapamycin or other treatments?
Yes, in culture, rapamycin definitely blocks that multiple axon phenotype. And we think it's due to regulation of expression of proteins such as SAT kinase.
Terrific.
Now, does this exactly happen in vivo? To be honest, we don't have proof of that. It's been very difficult to show multiple axon in Golgi staining, et cetera. It's very hard to get in one section multiple axons of the same neuron. It's a difficult experiment, at least for me. So we haven't shown that. We have shown, however, that if you're, one sort of pseudo correlate of that are these migrating neurons with multiple leading processes, which some people sort of think of as polarity abnormality in early developing cerebral cortex. So we have that abnormality in vivo, and rapamycin treatment seems to block that abnormality as well.
Actually, on the heels of that question, are there any developmental effects in the exuberant neuron growth?
Developmental in what sense?
Do they happen, I mean, I guess you've really looked, where in development have you looked? Is it all early? You know, at certain points in development, there's more growth, and then there's pruning in other.
Oh, I see what you mean. Yeah, we have not looked at pruning very much. The retinal uniculate projections I was showing you were done at a time before pruning, so we don't think it's the pruning, but we think it's the aberrant projection formation. We're just starting to look at dendritic spine pruning with Bev Stevens, and we have a project on that, but we have not looked at pruning as in detail as we should.
Is it possible for a patient to have multiple tumor disorders like tuberous sclerosis plus NF, Turcot syndrome, Van Hippel-Lindau's, and things of that nature?
I mean, it's always possible. I think it's sort of lightning striking twice type of thing, but you have to really look at the clinical picture and determine whether there's more than one thing going on. I mean, we wouldn't have guessed that the patient we are taking care of would have Fragile X and TSC at the same time. As you can imagine, they are both rare disorders, but anything is possible.
So continuing on the environmental risk question, so you mentioned cerebral hemorrhage as a very strong predictor
Cerebellar
of
hemorrhage.
autism. Are there particular triggers for the cerebral hemorrhage that are the specific ones that lead to autism?
Not that we know of. I mean, in the newborns, we see hemorrhage, around the time of birth, relatively frequently in the ICU, in the neonatal ICU. The reason for those are not well understood. We often do the usual hematological workup to see if they have a predisposition to bleed, but those things are usually negative. We don't know if any underlying causes, I should say, but that was just to make sure. It's the cerebellar hemorrhage that seems to have the most effect. It's not the cerebellar hemorrhage, which we see a lot of. We see a lot of interventricular hemorrhage, for instance, and that has a potential increase in autism risk too, but not as much as cerebellar hemorrhage.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.